Okay, so like what I mentioned, we started talking last time about metabolism. So we already are aware as to what constitutes really metabolism, diba? We already agreed that metabolism is just the sum total of all the chemical reactions that are going on inside the cell. And because there's plenty of chemical reactions that are going on inside the cell, I actually designed your lesson that we will only focus on the chemical reactions that are involved in what we call the biochemical energy production. So as mentioned, biochemical energy production is the process in the cell wherein we're able to obtain the energy stored in the food that we eat into usable form of energy in the body. So these chemical energy in the food are stored in the chemical bonds. Therefore, biochemical energy production would require the breakdown of these chemical bonds. Therefore, biochemical energy production is basically catabolic, no? And then there are 4 stages of biochemical energy production, diba? Stage 1, which is digestion. Stage 2, which is the production of the acetyl group. And then stage 3 was the Krebs cycle or the citric acid cycle. And then we talk about the citric acid cycle in detail, okay? And then stage 4 is the electron transport chain, which we will be considering today, okay? So this is now showing you the summary of the Krebs cycle. So this is a very elegant cycle. And like what I mentioned, we need to memorize this cycle. No, this is non-negotiable. So you're going to memorize this cycle by heart, okay? So if we're going to interpret the cycle as shown in the screen, again, what is the fuel of the Krebs cycle? Or what starts the Krebs cycle? What substrate must need to enter the Krebs cycle in order to start it up? We need to have your fuel class. What's the fuel of the Krebs cycle? You have your acetyl-CoA, no, your acetyl coenzyme A, diba? So that starts up your Krebs cycle. So what are the byproducts? For every acetyl coenzyme A that enters the cycle, you get 2 carbon dioxide. Those are the mga important byproducts, no? For every acetyl coenzyme A that enters the Krebs cycle, always remember, the important byproducts that you need to remember is it produces 2 carbon dioxide, 3 NADH, 1 FADH2, and 1 GTP, which is chemically equivalent to 1 ATP molecule. Do you follow? So these are the important byproducts upon the complete oxidation of acetyl portion of the acetyl coenzyme A. Remember, class, what's the goal of the citric acid cycle? The goal of the citric acid cycle is to convert the carbons of the acetyl to what? It would need to oxidize the carbons of the acetyl to carbon dioxide. Do you follow? To 2 carbon dioxide. It's the goal of the citric acid cycle. So that is why in the end, you are producing the reduced coenzymes NADH and FADH2 because the entire process requires a lot of oxidative steps, okay? So upon the production of the NADH and FADH2, unsa gito lang na-gain, what did NADH and FADH2 gain in the process? They were able to gain what? No? What did NADH and FADH2 gain in the process, class? They gained, something na gain nila during oxidation. Okay, they were able to gain hydrogen and electrons. So the electrons now, no, so because NADH and FADH2 were able to gain electrons, so they are now already highly energetic. So they're already rich in energy. So they will then proceed to the stage 4. So always remember, only NADH and FADH2 can proceed to the stage 4 of biochemical energy production. And the stage 4 of biochemical energy production will be talked about in detail later on. Okay? So another thing you need to realize about the citric acid cycle, class, although we actually view it so far as a, we actually view thus far the citric acid cycle as a catabolic pathway. But in reality, class, the citric acid cycle can be viewed as an amphibolic pathway. When I say amphibolic pathway, meaning to say it can also be involved in catabolism. So that is why I'm telling you that citric acid cycle is a central pathway for metabolism. Okay, aside from being part of biochemical energy productions, the metabolites or the byproducts in citric acid cycle can also be utilized to form the other macromolecule. For example, as you could see there, diba? Oxaloacetate is one of the byproducts in the citric acid cycle. Am I right? So you would see there that oxaloacetate can go to another pathway where it is converted into carbohydrates. Do you follow? Or perhaps the other, for example, alpha ketoglutarate can also be converted to form your what? No? It can also be converted to form your other sugar, other carbohydrates, for example. Do you follow? It can be converted to glutamate, no? So, and the other things can also be, the other byproducts can also be converted to form the other macromolecule. So I do not intend to memorize this. I don't intend for you to memorize these things, but what I want to emphasize here is the fact that your Krebs cycle can also serve the anabolic pathways. And therefore, because it is both catabolic and anabolic, it can be classified as an amphibolic pathway. Okay? So that's why it's a very central pathway. Okay? Sige. So we're already done now with the citric acid cycle. Again, the important byproducts in the citric acid cycle that can proceed to the stage 4 is your NADH and your FADH2. Because again, your NADH already contains highly energetic electrons. So they were able to gain electrons due to the oxidative reactions in the citric acid cycle. So the electrons in the NADH and the FADH2 will then proceed to what we call the electron transport chain. So from the name itself, the purpose of the electron transport chain is to transport electrons, okay? So basically, class, when we talk about the electron transport chain or the ETC, it is simply a chain of proteins and coenzymes, no? So you would notice that there are chains and there are chains of proteins and coenzymes. So basically, the electrons that are gained by your NADH and your FADH2 will be passed on to the different proteins and coenzymes, no? So they will be passed on to these proteins. So as electrons move along this chain, it will be able to gain energy, no? Just like your electricity ba, if we're going to relate it to something we experience in the daily basis, for example, electricity, diba? Electricity is very energetic. In fact, it can actually fuel your air conditioning unit. It can fuel your other appliances, diba? So what is electricity? It is simply a flow of electrons. Do you follow? Therefore, if we relate it to the electron transport chain, therefore, no? So when electron flows in the electron transport chain, it is also able to gain energy. So the energy that is gained by the electron transport chain is used to produce ATP in another process we know as oxidative phosphorylation. So the energy that is gained here will be utilized to produce ATP in oxidative phosphorylation. Okay? So that's it. No? So ang goal ng electron transport chain, class, the electrons will move along the chain and then the final electron acceptor will be oxygen. So meaning to say, so for example, no, gikan sa citric acid cycle, ang NADH iyang ipas ang iyang electrons ngadto sa electron transport. So that electrons will transport and then afterwards, it will then proceed to the oxygen in the mitochondrial matrix because your mitochondrial matrix contains a lot of oxygen, man. So once the electrons reaches the oxygen in the mitochondrial matrix, it will then be converted to water. Therefore, electron transport is considered to be aerobic process, because this is where we see that oxygen takes part in the reaction. Oxygen here plays the final acceptor of the electrons. So this is the final acceptor of the electrons. Is that clear? Is that clear? So that's the summary of the electron transport chain. So the electron will travel in the chains of proteins and coenzyme until eventually it will reach the oxygen gas in the mitochondrial matrix, converting it to water. So again, this is aerobic because of that process. Okay, it's an aerobic process. Again, electron transport chains, class, no? The proteins in the electron transport chains are all situated in the inner mitochondrial membrane. So meaning to say, if I have a mitochondrion here, so my Krebs cycle, of course, here. This is where my Krebs cycle here. So ang akong electron transport chain, they are located in the inner mitochondrial membrane. Okay? They are located there in the inner mitochondrial membrane. So the NADH and the FADH2 that is produced in the matrix will then proceed to the electron transport chain located in the inner mitochondrial membrane. Okay? So that's it. That's the story of your electron transport chain. Okay, so let's talk about this in detail one by one. So by the way, electron transport chain, by the way, class, no, is actually composed of 4 complexes, no? As you could see there, it comprises 4 complexes. So it's called complex 1, complex 2, complex 3, and complex 4. Okay? Sige. Again, like what I mentioned, it is highly it is highly aerobic. In fact, 95% of the oxygen in the body is used in the ETC. So that is why if there are processes of, or if there are disturbances in the intake of oxygen in the body, then that could always destroy the electron transport chain, because this needs a lot of energy. This needs a lot of oxygen. Okay, so again, like what I've mentioned, electron transport chain comprises 4 complexes, no? So it has complex 1, which is called NADH coenzyme Q reductase, and then the complex 2, which is also known as your succinate coenzyme Q reductase. Complex 2 is known as your coenzyme Q cytochrome reductase. And then complex 4, the last complex, is called cytochrome oxidase. Okay? Aside from the different, by the way, the different complexes class comprises proteins, no? The different complexes comprises proteins. So aside from the 4 complexes, your electron transport chain also have what we call mobile electron carriers. So anong gitawag man sila nga mobile electron carriers? Because, you know, as you could see there, the different complexes are separated from each other. So it's actually the responsibility of these electron carriers to carry electrons from one complex to another. So that's why they're called 2 mobile electron carriers. So the 2 mobile electron carriers are your coenzyme Q, or also known as ubiquinone, and cytochrome C. Okay, so I will explain later the significance of these mobile electron carriers later on. But I want you to emphasize that their main function is for them to actually transport electrons from one complex to another. Because again, the 4 complexes are separated from each other. Okay, so let's now talk, let's go to the different complexes one by one. Okay, let's start with complex 1. So complex 1 is known as NADH coenzyme Q reductase. So what happens in complex 1? Okay, remember, what are the 2 reduced coenzymes that are produced in the Krebs cycle? What are the 2 reduced coenzymes produced in the Krebs cycle? You have your, what's the reduced coenzyme na produce a Krebs cycle, class? You have your NADH and your FADH2. Okay. So when you talk about the complex one, this is where this is the entrance of the complex one. This is the entrance of NADH. Do you follow? So in other words, there are two types of reduced coenzymes produced in a Krebs cycle, but they enter the electron transport chain in different complex. So complex one is where the electrons of the NADH will enter. That's why it's called NADH CoQ reductase, to indicate that it is where the electrons of your NADH will enter. So the electrons of NADH will enter the complex one. So the process is it's actually comprises of FMN and FESP. So what do we mean by this? FMN stands for flavin mononucleotide, and then FES is iron sulfur protein. Okay, so muna siyang process class, no? So upon reaching the complex one, the electrons will first be transferred to your flavin mononucleotide. Afterwards, the electrons will then be transported to your iron sulfur protein. So di ba electron transport, it's not very complicated. The electrons are just being passed on from one component of the complex to another component. So the order is from NADH, it will be passed on to flavin mononucleotide, and then the electrons that is gained by FMN will then be passed on to iron sulfur protein. So the final acceptor of electron in your complex one is your coenzyme Q. So your NADH, so that's why it's called NADH CoQ reductase, to indicate that the final acceptor of electrons coming from NADH in complex one is your coenzyme Q. Remember, class, your coenzyme Q is an electron carrier. So that means to say that it will be the one that carries the electron from complex one to the next complex. Do you follow? Do you follow? So maura na siya ang progression. Electrons coming from NADH will be passed on to flavin mononucleotide. Flavin mononucleotide will then pass it on to iron sulfur proteins, and then the iron sulfur proteins will then pass it on to your coenzyme Q. So where will coenzyme Q pass the electrons? So will it pass to complex 2 or to complex 3? So again, it will pass it on to complex 3. Okay, so the order is NADH will enter complex 1, but eventually upon its as its electron is passed on to the coenzyme Q, it will then be passed on to complex 3. Dili siya paingon sa complex 2. Although the name is very ambiguous ba. It tells us that after complex 1, it should be complex 2. No. Again, always remember, so the electrons, as it's gained by the coenzyme Q, will then be passed on to complex 3. So that's the next steps of the electrons for your NADH. Okay? Am I making sense? Am I making sense, class? Hello? Am I making sense? Okay. Sige. So let's now proceed to complex 2. So the reason why complex 2 class is not the next step for complex 1, because your complex 2 is the entry point of your FADH2. Okay? So it's the entry point of FADH2. So your complex 2 is a very, very simple class, no? It's actually a very, very simple complex. In fact, unlike your complex 1, it is only comprising of iron sulfur proteins. Therefore, the electrons coming from FADH2 will be transported first into the iron sulfur proteins of complex 2. Afterwards, just like your complex 1, the final electron acceptor of complex 2 is CoQ reductase, CoQ, coenzyme Q. So therefore, The coenzyme Q now will then transport it to complex 3. So the final, so mean to say ba, both the electrons coming from NADH and FADH2 will enter at separate side of the electron transport chain. But eventually, both of their electron will be carried by coenzyme Q to complex 3. Do you follow? Do you follow? Am I making sense? So kung baga class, if you're going to put that into perspective, also, I think it would be better if I draw it myself. So for example, if this is my complex 1, what will enter here? What will enter here? My NADH. So my NADH transports the electrons. It's reconverted to NAD+. So asa mo tong electrons? Una mo agi. The electrons will first pass on to your FMN. And then FMN pass it on to iron sulfur proteins. And the iron sulfur proteins will pass it on to your coenzyme Q. Okay? And then, so you have your complex 2. So what enters in complex 2 class? What enters in complex 2? So what enters in your complex 2 is your FADH2. So as your FADH2, as the electrons are passed on, it's converted back into FAD. Okay? So what is present in your class? What is present in your complex 2? It only comprises iron, sulfur protein. So the electrons will pass in there and then it will pass to coenzyme Q. So afterwards, the coenzyme Q will pass their electrons now to your complex 3. Is that clear? Is that clear? So that's it. No, it's just passing of electrons. They follow. It's very, very complicated. All right? Sige. So that's it. No, so we're already, so upon so ang complex 1 or complex 2 entry point na siya, no? It's just the entry point of the NADH and the FADH2. But basically, they will all be transported, their electrons will all be transported to coenzyme Q. No? Okay. By the way, coenzyme Q is also known as, again, it's also known as ubiquinone. The other name for coenzyme Q is ubiquinone. The reason why it's called ubiquinone class, because it's ubiquitous. It's an ubiquitous kind of coenzyme. Why did I say it's ubiquitous? Because most of the cell contains ubiquinone. No, it's very, very abundant, kumbaga. So in any parts of the cell, you would see coenzyme Q. That's why it's called ubiquinone. Okay. So afterwards, so what happens now in complex 3? So the complex 3 will be the one that accepts now the electrons coming from complex 1 and complex 2. So the coenzyme Q carries now the electrons from NADH and FADH2 towards your complex 3. So your complex 3 class comprises what we call cytochrome. No? It comprises what we call cytochrome. So cytochromes are compounds containing a heme. So this is a heme. This is the structure of a heme. So what is a heme class? Where did we first encounter the heme? We first encountered it in the chemistry of your hemoglobin. Am I right? When we studied about hemoglobin, the heme is actually the prosthetic group of your hemoglobin. Am I right? So your complex 3, which actually contains cytochrome, also contains a heme. Do you follow? So what's now the sequence of electron transport in your complex 3? So, di ba, kanina na nag-carry sa electrons, no? So your coenzyme Q is the one that carries now the electrons, correct? So ang first niyang pasahan is your iron. So just like the other proteins, it also just like the other complexes, it also contains iron sulfur proteins. So the electrons coming from coenzyme Q will first go to iron sulfur proteins, and then the iron sulfur proteins will pass it on to cytochrome B. So cytochrome B will pass it on again to iron sulfur protein, and then iron sulfur protein will pass it on again to cytochrome C1, and then eventually it will pass it on to cytochrome C. So the final acceptor of electrons in complex 3 is your cytochrome C. And as we know, cytochrome C is also an electron carrier. Do you follow? So just like your coenzyme Q, your cytochrome C is also an electron carrier. So the cytochrome C now will be the one that carries the electron towards complex 4, the last complex of the electron transport chain. Do you follow? Do you follow? Am I making sense, class? Am I making sense? Hello, do you get that?
Yes, sir.
Okay, so that's your complex 3. Okay, so again, no, natay duha ka electron carrier, no, we have CoQ and cytochrome C. So what did you notice, class? Based on the illustration of the electron transport here, what did you notice about CoQ and cytochrome C? Try to look at them. What is odd between them? Try to look at their direction of electron transport. What could you notice? Okay, so where is coenzyme Q located and where is cytochrome C located? What did you notice there?
On both ends, sir. Nas la paggawas.
Negawas ba si coenzyme Q? Okay, so as you can see, so coenzyme Q is actually present only in the membrane, diba? This is your membrane man, correct? Diba? So it is actually present in the inner membrane. So diba, remember class, if you have a membrane, this is actually, unsa man nga area diri, if you have a membrane, this area here, is it polar or non-polar? The inner part of your membrane, that's non-polar, correct? Therefore, your coenzyme Q moves in the non-polar portion of the membrane. The reason for that is your coenzyme Q is highly non-polar. That's why the way it carries electron is lateral direction in the membrane. Do you follow? Do you follow? So how about your cytochrome C? What did you notice about cytochrome C? Was it present inside the membrane or outside the membrane? Outside. Therefore, it is polar. No, it is polar. It is water soluble. In fact, always remember, cytochrome C is the only water soluble cytochrome because there's plenty of cytochrome. Oh, naman kay cytochrome B, na kay cytochrome C1, correct? But only cytochrome C is soluble in water. Do you follow? So that's why the way it carries electron is outside the membrane, in the polar portion of the inner mitochondrial membrane. Okay, so that's it. Okay, I just like to emphasize that. But after reaching complex 3, you are already passing on the electron to cytochrome C. So cytochrome C will then transport it to complex 4. So in complex 4, it actually comprises, no? It actually comprises as well cytochromes, okay? But unlike your other cytochromes, your cytochromes class actually contains copper. Do you follow? Aside from cytochromes, no? Your complex 4 also contains copper, okay? So cytochrome A contains copper aside from iron. Okay, ang original na cytochrome class only contains iron man. But in the case of the cytochromes present in your complex 4, it additionally contains copper. Okay, so what happens here? So the electrons coming from cytochrome C will be transported first to cytochrome A. So the order is, no, so cytochrome A contains copper and iron. So the electrons from cytochrome C will first be transported to the copper of cytochrome A, and then to its iron, and then to cytochrome A3. So the final acceptor of electrons in complex 4, as mentioned, is your oxygen. So upon the acceptance of electrons in your oxygen, it will then be converted to water. So in this case, your electron transport is already done. Okay? So sa kahaba-haba man ang posisyon, so its only goal is to transport the electrons coming from your NADH and FADH2 to oxygen. So as a summary, so this is the process, no? So this is the process of your electron transport. So again, you have complex 1. So what enters in complex 1, class? You have your NADH. So upon its entry, it's converted to NAD plus again. So the electrons are first passed on to FMN, and then FMN pass it on to iron sulfur proteins, and then to CoQ. So you have complex 2. What enters in complex 2? You have your FADH2. So FADH2 is converted back to FAD. And then the electrons are passed on to iron sulfur proteins and then to CoQ. Okay? And then CoQ will pass it on to your complex 3. Correct? So what is in complex 3? Kinsa may unang receive? The first to receive will be your, what is first to receive? Your cytochrome. What cytochrome? Cytochrome B. Followed by cytochrome C1. And then the last acceptor is your cytochrome C. Am I right? And then cytochrome C will then pass it on to your, complex 4. So in complex 4, kesa may mo first accept. It is first accepted by your cytochrome again, but it's cytochrome A, and then cytochrome A3. Am I right? Cytochrome A3, sunod class, no? And then followed by the final acceptor is your oxygen, converting it to water. Okay? So that's the story of your electron transport. So again, it's not very difficult to understand, because just like what I mentioned, it's just the electrons are moving in these different complexes. Now the question is, did we already produce ATP here, class? Did we already produce ATP here? Yes or no? Did we already produce ATP? Not yet. Diba? Not yet. But then again, always remember, so aside from being electron transport, no? Another function of the different complexes, class, is they pump hydrogen ions. Always remember, another function of the different complexes is they are able to pump hydrogen ions. So as the electrons move along these different complexes, they will be able to gain energy. So the energy that they gain will be able to, kay membrane, di ba? Naman sila sa membrane, class, no? They are present in the membrane, man. So this is the matrix. This is the intermembrane space. So as the electrons move along, no? As the electrons move along the different complexes, they are able to gain enough energy to pump hydrogen ions from the matrix into the intermembrane space. So si complex 1, makapump na siya 4. Si complex 3, makapump na siya 2. And then si complex 4, makapump of 4. So only complex 2 is not capable of pumping hydrogen ions. So that is now the other consequence of your electron transport. Aside from the fact that it is where the electrons move along, always remember, you need to also realize that as electrons move along in the complex, they gained enough energy in order for them to pump hydrogen ions coming from the matrix into the intermembrane space. So this pumping of hydrogen ions from the matrix into the intermembrane space is the key factor as to how it is able to produce ATP. So why am I saying this? Okay, so let me explain now to you the next stage of stage 4. So the next stage of step 4 is called as your oxidative phosphorylation. So like what I've mentioned, your complexes will pump hydrogen into the intermembrane space. So what happens, no? So oxidative phosphorylation class is based on what we called, a theory we called chemismotic theory. This was first elucidated by Peter Mitchell. So the biochemist who first elucidated the chemismotic theory is Peter Mitchell. So what is the significance of pumping hydrogen ions into the intermembrane space? Okay, so as you do Krebs cycle, there will be plenty of NADH and FADH2. Am I right? So therefore, there will be a lot of electrons that will move along this complex, correct? So as there are a lot of electrons that moves along the complex, you would expect that there will be plenty of hydrogen ions being pumped, no, into the intermembrane space. So what happens, no? So what happens when you pump a lot of hydrogen ions into the intermembrane space? What happens now? There will be an increase in hydrogen ions here in the intermembrane space. However, in the matrix, there will be a decrease. Do you follow? There will be a decrease. So meaning to say, this difference in concentration creates what we call a proton gradient. When we say proton gradient, you have a membrane, and then one side of the membrane has high concentration of hydrogen. The other side of the membrane contains low concentration of hydrogen. This is now the consequence of your electron transport chain, because it pumps a lot of hydrogen ions to the intermembrane space, causing for it to accumulate a lot of hydrogen ion. So what happens is, it will go, because there's already plenty of hydrogen ions in the intermembrane space, no? Because there's already an increase of hydrogen ions here in the intermembrane. So the hydrogen ion here that is already very abundant in the intermembrane space would want to go back to the matrix. Maugay gitawag sa mga chemismotic plus osmotic, mean to say from higher concentration to lower concentration of movement. So because there is a higher concentration already of hydrogen ions in the intermembrane space, it would want to go into the matrix again. No? Am I making sense? Am I making sense? So where will it go back? No, it will go back in what we call the ATP synthase. So this is where the entry, no, re-entry. So again, this is where this is the re-entry of hydrogen ion, your ATP synthase. Okay, so that is where the hydrogen ions will enter. So therefore, upon the entry of your hydrogen ions class, no? So ang kanang mong ATP synthase, it look like this. Your ATP synthase looks like this. So it looks like that. So in the bulb of your ATP synthase, na ADP of phosphate attached. Do you follow? It has an ADP and phosphate attached there. So as hydrogen ion re-enters, no? As hydrogen ions re-enters the ATP synthase, what happens? The bulb of the ATP synthase will rotate. So once the bulb of the ATP synthase will rotate, ADP will combine with phosphate, forming your ATP. So that is the reason why we are able to produce ATP in the end. And it is fueled by the proton gradient that is actually produced as electrons move along in the electron transport chain. Am I making sense? Am I making sense? Class, kasabot rataan ni? No, so this is oxidative phosphorylation. It's coupled to the electron transport chain. Gitawag siya nga oxidative phosphorylation because the energy that drives the production of ATP kay gikan man sa oxidative reaction. Gikan ba siya class sa NADH ug FADH2? Am I right? So asa man na gikan si NADH ug FADH2? Gikan ba na sila og oxidation? That's why it's called oxidative phosphorylation, to indicate that the energy required or the energy that is used to produce ATP at ATP synthase is what causes the production, is coming from oxidative process. Do you follow? Do you follow? Class, am I making sense here? No? Yes. So yeah, as electrons, it's not pump. Electrons are not pumped, huh? As electrons move along, no? The electron transport chain, it allows for hydrogen to be pumped into the intermembrane space. Okay, so I will again illustrate it in my own. So diba? So asa gani na na-locate ang atoang kuan class? Asa na na-locate? So atong i-drawing ang mitochondrion, ha? Kay para maka-understand mo. So mo bitaw na giingnan ta mo nga you need to memorize the anatomy of mitochondrion. Where is the electron transport chain located? Electron transport chain is located in the where? Where is asa nga mitochondrial membrane? What specific? Doon ka man ka mitochondrial membrane, outer or inner? Inner. So dire. Diba? Arang na siya na-locate. Right? So ato na siyang i-zoom in. Diba? I-zoom in na nato. So ilan na siya. So this is now your inner membrane. So in the inner membrane, you got there complex 1. Naakay complex 2. Naakay complex 3. And you also have complex 4. And then you have here your enzyme, kaning murag bulb. You have your, this is called your ATP synthase. Okay. So this part, di ba, kung inner membrane, what is present, what is being separated by the inner membrane class, di ba? The inner part of the inner membrane, so may patawag ani diring dapita. This is your matrix, so dire, matrix na. How about here? How about this portion? This is your intermembrane space. Okay? Sige. So ang electrons from NADH, di na ako i-detail class ha, musulod dire sa imong complex 1. And then, moadto sa complex 3. Ang electrons ni FADH2 enters complex 2, goes to complex 3. And then complex 3, the electrons moves to complex 4 until eventually oxygen is converted to water. So the movement of electrons in the different complexes causes for the pumping of hydrogen ions, specifically in complex 4, complex 3, and complex 1. No? So as electrons move along the electron transport chain, Complex 1 will pump 4 electrons. Complex 3 will also pump 4 electrons. And then lastly, complex 4 will pump, complex 3 will only pump 2 electrons, 2 hydrogen, no? And then your complex 4 will pump 4 hydrogen ions. No? So from the matrix going into the intermembrane space. So because the electron transport chain is always operating, kay sige man tag naay NADH. So the electrons always move along the chain. Therefore, there will be, what happens now, if you keep on pumping hydrogen ions in the intermembrane space, asa man karon, so there will be a proton gradient wherein there is a difference between the concentration of hydrogen in the matrix and in the intermembrane space. So there will be high H plus and low H plus here. So that's called a proton gradient when there's a difference between the hydrogen ion concentration between them. So what happens? Due to osmosis, the hydrogen ions in the intermembrane space would want to go back to the matrix. Where will it go back? Where will it go back, class? It will go back at what specific area will it go back? Okay, it will go back in the ATP synthase. So as the electrons, as the hydrogen ions goes back to the matrix through the ATP synthase, what happens? The ATP synthase will rotate. So as the ATP synthase will rotate, it will cause the combination of ADP to ATP. So that's it. No? It is able to produce now your ATP. No, this process would not occur in the absence of oxygen because again, you need to have an oxygen to complete the electron transport. Because like what I've mentioned, the final acceptor of oxygen is the final acceptor of electrons in the electron transport is always your oxygen. So it cannot occur in the absence of oxygen. Okay, if wala oxygen dili, asa man paingon ang electrons ni complex 4? It has no final acceptor. Therefore, there will be a disruption in the electron transport. So in order for this process to occur efficiently, the passing of electrons must occur in this manner. So that is why if there is a substance that can say inhibit the amount of oxygen or that can lower the amount of oxygen, that can be very detrimental to your electron transport. Do you follow? It can be detrimental to your electron transport because this is very much requiring the presence of oxygen. Is that clear? In fact, again, 95% of the oxygen of the body is utilized by your electron transport chain. Okay? So that's why this is highly aerobic process. Class, am I making sense? Kasabot rataan nga process? Do we get this? Class, am I making sense here? Okay, so this is now how we are able to produce ATP. Okay, so how much ATP did we produce after all in all? Okay, remember, no, so remember, complex 1, bali dito class, sorry. I made it to reverse the I. Complex 1, sorry. Complex 1 pumps 4 hydrogen ion. Complex 3, they ang pump of 4. And then complex 4 pumps 2 hydrogen ions. So if this is the entry, no? So NADH, where will NADH enter? NADH enters at where? Complex 1. So in the end, the electrons of NADH, how many hydrogen ions is it able to produce? Or is it able to pump? How many hydrogen ions can NADH pump? So 4, di mo ari siya sa complex 3 plus 4, and then aside plus 2, so it's a total of 10. Do you follow? Do you follow? Kay mo move along man siya class, dili man na siya mo stay ra sa complex 1. Am I right? Am I right? Do you get what I meant? So the electrons of NADH, all in all, is able to pump 10 hydrogen ions. How about your FADH2? Your FADH2 enters here. So how many electrons it is able to pump only? What many hydrogen di ka? Diba? So it will only pump 4 and then 2. So a total of 6 electrons. Do you follow? Do you follow? Class, am I making sense here? Okay. So what is the significance of knowing this? So we have the concept what we call PO ratio. So what's a PO ratio? So a PO ratio is the number of molecules ATP that is formed for every electron that flows in the electron transport chain. So according to studies, di ba, nakay ko ano, nakay ATP synthase. So the ATP synthase, every rotation of the ATP synthase would actually cause the production of your ATP. The question is, how many hydrogen ions will need to enter the ATP synthase in order for it to rotate? Do you follow? Kaya dili man na usara ka hydrogen ion class. So the question is, how many hydrogen ions will will enter the ATP synthase in order for it to rotate? So according to recent studies, according to recent studies, 4 hydrogen ions would need to enter the ATP synthase in order for it to produce a ATP. So mean to say, for every 4 hydrogen ions that re-enters the ATP synthase, it is able to produce ATP molecule. So therefore, NADH, pila man ka electrons, pila man ka hydrogen ions ang na-pump ni NADH class, di ba? We are able to pump 10 hydrogen ions, correct? Therefore, so if 1 ATP is produced per 4 hydrogen ions, then that tells us that NADH that enters ETC will be able to produce 2.5 ATP per 2 electrons. On the other hand, because FADH2 only was able to pump 6 hydrogen ions, so therefore, it is only able to produce 1.5 ATP per 2 electrons. Do you follow? Do you follow? So meaning to say, ang kanang NADH nga mo enter sa ATC, ang equivalent niya nga ATP kay 2.5. Ang kanang FADH2, for every FADH2 that enters the ETC, it is able to produce 1.5 ATP. So we can now calculate the amount of ATP that is produced starting from the Krebs cycle down to the electron transport chain. Remember, these are the steps that produces the NADH and the FADH2. So again, if NADH will already enter the ETC, it is able to produce 2.5 of ATP. If FADH2 will enter, it is able to produce 1.5. And remember, we also produce 1 GTP, which is chemically equivalent to 1 ATP. So 3 of NADH times 2.5, that's 7.5. 1 times 1.5, that's 1.5. And then 1 GTP. So meaning to say, for every acetyl-CoA that enters the Krebs cycle, and then it's producing NADH and FADH2 that will enter the ETC, it is able to produce 10 ATP. Do you follow? Do you follow the progression, class? Class, do you follow the progression of how we came up with the amount of ATP that is produced? Hello, ladies and gentlemen. How about the others? Did we all get this? How about the others? Did you get how we were able to come up with 10 ATP? Okay? So inani rin siya. So again, so di ba? Acetyl-CoA enters Krebs. So Krebs produces how many NADH? How many NADH that Krebs produces per acetyl-CoA? It produces 3.
3.
How many FADH2? 1. And then 1 ATP or GTP. So kani, this will proceed to ETC and oxidative phosphorylation. So how much ATP is produced per NADH in the ETC? This is able to produce 2.5 and this is able to produce 1.5. Am I right? So 3 times 2.5, that's 7.5. 1 times 1.5, that's 1.5. And then 1, so a total of 10 ATP. Do you follow? Do you follow? So ang katong carbon dioxide, kung di na ito kwenta ko, no? Kaya wala naman to siya ATP, no? It will be excreted naman. Do you follow? Do you follow? Class? Okay. So question so far? Question so far. So in the end, for every acetyl-CoA that enters the Krebs cycle, going to ETC and oxidative phosphorylation, we are able to produce 10 ATP. Okay? Sige. Questions? Wala? Ladies and gentlemen? No questions? Uy, asa naman mo? Okay, no questions so far. Sige, so if no questions, then I will give you 5 minutes break. Let's be back at 9:40. Okay?
Okay.
Yes, you have a question, Ravi?
Kato ganina sir ba sa complex 3 ingon ka mo ang CoQ mo una og pass sa ansa to site sa cytokines B
Yeah. Yeah. Diba? Right?
and then wala mo na-appeal ang iron.
Irons are, yeah. Wala ko na-appeal sa iron sulfur protein. So you're correct, no? So it would need to go to iron sulfur protein first before going to cytochrome B. I stand corrected about that. Kaya akong i-consider kaya mga cytochrome B ra. But yeah, there
Una ang iron.
there are also iron sulfur protein. Anyway, this is the order, no? So in your notes, I've written there the order of how the electrons are being passed on in the different complexes. Okay?
Okay, sir. Thank you. Okay.
Sige. Any other clarifications before we go to the break? So wala. So if not, then let's be back at 9:41. Okay? Okay, sige. So again, biochemical energy production starts by the food that we eat, and then it's being digested to their corresponding building blocks. And then building blocks are then converted to what? They're then converted to acetyl-CoA. Am I right? So this process is digestion. And then acetyl formation. So the acetyl-CoA, where does it enters? It enters now the stage 3. What is a stage 3 biochemical energy production? Acetyl-CoA enters the Krebs cycle. So where does the Krebs cycle occur? It occurs in the matrix. So what is produced during Krebs? It is able to produce, of course, 2 CO2, 3 NADH, and then 1 FADH2, and then 1 ATP. So the NADH and the FADH2 will proceed to the electron transport chain. Am I right? To the ETC, correct? So what happens to the ETC class? They will able to pump hydrogen ions, no? Able to pump hydrogen ions. So what happens? The hydrogen ion will then go back in the in the ATP synthase, causing for its rotation and then the production of its ATP. Am I right? to form ATP, not to form ATP at this point. Okay, so that's so far as we know. So we're already aware how acetyl-CoA can actually be utilized to form your adenosine triphosphate, which is now the energy currency of the cell. So the ATP that is produced in this process will then be transported to the different parts of the cell for usage, because hydrolysis of the phosphate group of ATP, diba ATP comprises, kuan man class, no? ATP comprises an adenosine, and then the adenosine is attached to 3-phosphate. Am I right? So when you break down even one of the phosphate group in your ATP, it will release energy. So the energy that is stored in ATP will be utilized by the different parts of the body for their different processes. However, so we already consider this, no? This is a common metabolic pathway. However, we haven't considered how did we reach into acetyl-CoA, diba? So when I have carbohydrates, for example, how did I become an acetyl-CoA in the first place? So let's say if I eat carbohydrates, how did it become an acetyl-CoA? So how did it even enter the common metabolic pathway? So that's the focus of my discussion now. We're going to elucidate how from food, coming from, for example, carbohydrates, how it is able to reach being an acetyl-CoA. Okay, so let's begin now with your carbohydrates. Okay, so stage 1 always begins with digestion. Again, digestion occurs in the digestive system. So here, I am showing you here the different stages of the digestion of carbohydrates. So digestion of carbohydrates, class, always begins in the mouth, no? So the saliva contains what we call salivary alpha amylase. So this enzyme, no? The enzyme salivary amylase, so when you eat carbohydrates, it is now able to hydrolyze carbohydrates, no? Specifically, what carbohydrates is being hydrolyzed by salivary amylase? Your starch. Do you follow? Your starch. So for example, I drink milk. What sugar is in milk? It contains lactose. So, or probably sucrose. And then I eat bread. What is in bread? It contains starch. So meaning to say, among these carbohydrates that you ingested, which undergoes chemical digestion in the mouth? Starch, lactose, sucrose. Only your starch. Do you follow? Because the enzyme amylase is only capable of digesting starch. Okay? So therefore, lactose and sucrose and other disaccharides goes unaffected. Okay? However, the digestion of starch by salivary amylase is short. It's actually cut short. Why? Because you would need to swallow your food. Am I right? So upon swallowing, the contents in the mouth will proceed to the stomach. What's the property of the stomach? Your stomach is acidic. Do you follow? So the acidic environment of the stomach inactivates the alpha amylase, no? It inactivates the action of your alpha amylase. That's why nga, upon reaching, no, as carbohydrates reaches the stomach, the gastric juices that is released by the stomach has no net effect on carbohydrate digestion. So that's why nga, di ba, your food will stay longer in the stomach, correct? So that is why if you eat a lot of carbohydrates, you feel full. Am I right? You feel full for a long time. Why? Because upon reaching the stomach, dili man siya ma-digested. So that's why you feel full when you eat a lot of carbohydrates. So after staying a long time in the stomach, it will then proceed to the small intestine. So in the small intestine, what happens? No? The pancreas will release bicarbonate. So what's the action of your bicarbonate? It will neutralize the content of the stomach. Therefore, upon reaching the small intestine, the contents, the acidic contents coming from the stomachs are neutralized by the bicarbonate secreted by the pancreas. And at the same time, in the small intestine, there is also the release of what we call pancreatic amylase. Therefore, in the small intestine, the digestion of carbohydrate continues until such time that all starch is converted to maltose. So at this point, we see now that we have mixture of disaccharides. So these disaccharides will then proceed to the intestinal mucosal cells. Because in the intestinal mucosal cells, we have your disaccharidase. So the disaccharidase will break down your disaccharides into monosaccharides. Do you follow? So once the monosaccharides are already produced, it is now able to be absorbed in the villi through active transport. So the villi will then transport the monosaccharide into the bloodstream. So the blood will then transport the different monosaccharides to the different parts of the body to enter the cell. So in the cell, this is where the metabolism of your carbohydrate will begin. Do you follow? Do you follow? Class, do we get the digestion of carbohydrate? So again, carbohydrate is absorbed in the intestinal lining, the villi, through active transport, where it is already now being transported to the different cells of the body. So once your carbohydrate or once your monosaccharide will enter the cell, it will then undergo the process known as glycolysis. So glycolysis is the main catabolic pathway of carbohydrate. So this connects the link now as to how we are able to convert carbohydrate into acetyl-CoA. Do you follow? So in other words, this is the link as to how carbohydrate can now enter the Krebs cycle. Do you follow? No? So this is the link. It's called glycolysis. From the name itself, it comprises the word glyco and lysis. Lysis means to break down. Glyco means sugar. Therefore, glycolysis is the breakdown of sugar. Its other name is known as the Emgen-Mayer-Hoff-Parnas pathway, to give credit to those biochemists who studied the elucidation of this pathway. In fact, glycolysis is the first pathway to be elucidated. So the first pathway to be discovered is your glycolysis. So unlike the Krebs cycle and the electron transport chain, glycolysis occurs in the cytoplasm or in the cytosol. No, it does not occur inside the mitochondrion. This occurs in the cytoplasm of the cell. So your glycolysis is very, very simple, class. Its goal is to convert glucose containing 6 carbons into 2-pyruvate containing now 3 carbons. That's the goal of your glycolysis. But in the end, it will be able to produce a byproduct of 2 ATP and 2 NADH. Do you follow? So in other words, even before reaching the common metabolic pathway, glycolysis is already able to produce ATP. Do you follow? Do you follow? Okay. Nakita man nato kaganiha kung asa ta nakaproduce of ATP. We're able to produce ATP once we already reach the electron transport chain and oxidative phosphorylation. But in the case of your carbohydrates, when it undergoes glycolysis, even before reaching ETC, it is already able to produce ATP. So the ATP that is produced, class, in your glycolysis is very important for other cells because your other cells contains no mitochondrion. There are cells in the body that contain no mitochondrion. For example, red blood cell does not have a mitochondrion. Therefore, it is not capable of doing electron transport chain. Am I right? Therefore, it would only rely energy from the ATP that is produced by glycolysis. Am I making sense? Am I making sense? Okay, even before reaching mitochondrion, nakaproduce naman ka of ATP. So that is why this is very important, no? For those cells that do not have mitochondrion. And a good example for that would be your red blood cell, because your red blood cell does not contain mitochondrion. Okay? Do you follow? Do you follow, class? Am I making sense? No? So that's the story. That's now the story of your glycolysis. So it occurs in 10 steps, no? It occurs in a 10-step process. And like I mentioned, I don't require you to memorize, no, the 10 steps of glycolysis because it's very, very impossible now to do so, especially that we no longer have the benefit of the time. So in our discussion of glycolysis, we will only focus on the chemical reactions or the steps that are very important in energy production. Okay, so this is a summary of the production of glycolysis. So for every glucose that enters the glycolytic pathway, no? For every glucose that enters the glycolytic pathway, you're able to produce 2 pyruvate, 2 NADH, and 2 ATP. Is that clear? So you're able to produce 2 pyruvate, 2 NADH, and 2 ATP. Okay? So again, I will not go to the reaction in detail in order for you not to be very stressed already. But I want to emphasize that glycolysis again is 10 steps. Step 1 to 5 is known as the energy investment, while steps 6 to 10 is known as the energy recovery. So what do I mean by this? Okay, try to look at step 1 to step 5, class. Any steps. You would notice that in order for the steps to occur, you would need to add a phosphate group to glucose, correct? No, you would need to add phosphate group to glucose. So where do we get the phosphate that we add in these steps? We get this from ATP. So that's why steps 1 to 5 is known as a energy investment phase. Because as you could see there, you were able to utilize a total of 2 ATP molecule. Do you follow? To phosphorylate your glucose. Okay. Kailangan man ta nga ma-biphosphate siya. Do you follow? So at the end of the step 5, your glucose was then broken down into 2, glyceraldehyde and dihydroxyacetone phosphate. So at this point, you would notice that this reaction starting from step 6 will then be able to pay off the ATP that is invested in the first stage of glycolysis. Okay? Sige, so what are the important steps in glycolysis? One important step in glycolysis class is step 3. Step 3 is what commits glucose to glycolysis. Do you follow? It is what commits glucose to glycolysis. Ano all committed, no? Anyway, so what I'm trying to emphasize here, so diba? Glucose, paangon is step 2. So once it already reached step 3, it no longer has a choice but to continue the glycolytic pathway. Do you follow? Kaya kung ari pa ka sa step 1 o step 2 class, wala pa man kay assurance nga kani siya kay mo continue sa glycolysis because these products, no? These products are branched pathway. Am I making sense? The products in steps 1 and 2 are branched pathway, meaning to say they can undergo different pathways. But however, if you successfully convert this product into this product in step 3, this product is already highly specific only for glycolysis. That's why I said that step 3 commits glucose to glycolysis because you can no longer proceed to any other reaction except the remaining reactions of glycolysis. Do you follow? So at which reaction is ATP produced? ATP, I mean ATP used, no? ATP is used at step 1 and step 3. Do you follow? ATP is used at step 1 and step 3. Okay? Do you follow? Do you follow? Okay, ang step 1 class is to phosphorylate your glucose. And then again, step 3 is again another phosphorylation. So you would need another ATP. So that is why nga. So again, when you produce fructose 1,6-bisphosphate, this fructose 1,6-bisphosphate is already what commits glucose to glycolysis because you can no longer be synthesized into something else. Do you follow? So after the class, after fructose 1,6-bisphosphate is produced, it will now cleave into 2 3-carbon containing compounds. So there are 3 carbon containing compounds that is produced in this process in reaction 4. So by the way, the cleavage here is asymmetric. What do I mean by this? When the fructose 1,6-bisphosphate is divided into 2, it's asymmetric. Meaning to say, the product that you were able to produce is, the product that you were able to produce are completely different from each other. You were able to produce dihydroxyacetone phosphate of glyceraldehyde 3-phosphate. Do you follow? So that's why I say it's asymmetric. Is that clear? So among the product that is produced in step 4 class, no? Always remember, between the products produced in step 4, only glyceraldehyde 3-phosphate can proceed to glycolysis. Do you follow? So duha man kabuo yung product. It can produce dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So only glyceraldehyde 3-phosphate can proceed to the reaction. Therefore, step 5 would need to convert dihydroxyacetone to another glyceraldehyde 3-phosphate. So in the end, what I'm trying to emphasize in the end of stage 5, it's as if there are 2 glyceraldehyde 3-phosphate that continues the glycolytic pathway. Do you follow? Class, do you follow? Am I making sense that there are two glyceraldehyde 3-phosphate that continues the glycolytic pathway? Am I making sense? Hello? Hello? Yes.
Can you repeat why there's two glyceraldehydes? Phosphate.
Okay. Sige. So again, reaction 4 of the glycolysis cleaves, no, di ba? You end up with fructose 1,6-bisphosphate. So it is being cleaved asymmetrically. When I say asymmetric cleavage, you produce two types of product. So the first product is your dihydroxyacetone phosphate. The next product is your glyceraldehyde 3-phosphate. Do you follow? So only glyceraldehyde 3-phosphate can proceed to glycolysis. So this dihydroxyacetone cannot proceed. Do you follow? So that is why there is the requirement of reaction 5. Because in reaction 5, the dihydroxyacetone phosphate that was produced originally in stage 4 is also converted to glyceraldehyde 3-phosphate. So that's why there are two glyceraldehyde 3-phosphate that enters. The one that is a product of step 4 and the one that is a product of step 5. Am I making sense? Am I making sense? No?
Yes, sir.
Oh?
Thank you.
Okay. Pag-cleave sa step 4, you already produce kuan man, 1 glyceraldehyde 3-phosphate. But in stage 5, this is converted now to your glyceraldehyde 3-phosphate. So in the end, you got 2 glyceraldehyde 3-phosphate entering the glycolytic pathway. So whatever byproduct we produce here, it's already times 2. Do you follow? Do you follow? Kasabot na class, whatever byproduct that is produced in step 6 to 10 is already times 2. Because we already agreed that there are 2 of your glyceraldehyde that enters the glycolytic pathway, that continues the glycolytic pathway. Do you follow? Do you follow? Class, am I making sense?
Yeah.
Okay. So what are now the important steps that has important byproduct? Step 6 produces NADH. Step 7 and step 10 produces ATP. Okay? Muna siya inyong timanan. So at step 6, you produce 2 NADH. At step 7, you produce 2 ATP. So that is why step 7 is known as the payoff step. Why is it known as a payoff step? Because this is the area wherein we saw that the 2 ATP that was previously used in the stage 1 is being paid. Do you follow? In other words, this is known as the break-even step. Kanang igo ra ka nakabawi sa imuhang investment. Correct? So initially, you invested 2 ATP. Correct? So at step 7, you saw that it is able to produce 2 ATP molecule. So you already paid off, or it's already break even to the ATP that you invested in steps 1 and step 3. Do you follow? So step 10 is where you see the net yield of ATP. So it is also able to produce 2 ATP here. So that's why I was able to mention to you that at the end of the glycolytic pathway, you are able to produce 2 ATP. And that is by virtue of step 10. Do you follow? Do you follow? Class, am I making sense here?
Yes,
Am
sir.
I making sense? No? Sige, kasabot ra ta ani. How about for others? No, so I am not expecting you to go to each step one by one, like what I mentioned, because it would already be very taxing for you. So what I want you to remember really are the important steps in the glycolytic pathway. So in the end, you would notice that you produce 2 ATP, 2 NADH, and 2 pyruvate. Do you follow? Do you follow? So inanin ni sa class O, inanin ni sa ba, no? Ang glycolysis. So you start with glucose. Okay. And glucose was eventually converted to fructose 1,6-bisphosphate. Pila ni ka steps dire? Pila ka steps to reach this point? We need 3 steps. And there is a concomitant use of 2 ATP just to reach the production of fructose 1,6-bisphosphate. So afterwards, fructose 1,6-bisphosphate is actually cleaved into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So only glyceraldehyde can proceed to glycolysis. Am I right? No? So therefore, what do we need to do first? We need to convert DHAP to glyceraldehyde 3-phosphate. So therefore, it's as if there are 2 glyceraldehyde 3-phosphate that proceeds to glycolysis. So step 6, you produce 1 NADH. So how many NADH do we produce? So if step 6 produces 1 NADH, so we produce a total of 2 NADH. They follow? Because again, 2 glyceraldehyde 3-phosphate naman. We already agreed that there are 2 glyceraldehyde 3-phosphate have already been produced. At step 7, you're able to produce 2 ATP as well. And then at step 10, you produce 2 ATP. So at the end of these steps, you're able to produce 2 pyruvate. So step 6, step 7, and step. So that's just we're going to understand na lang class, no? The glycolytic pathway. Focused only in the salient features of glycolysis. Kay para dili na mo magsigig memorize sa each step. Kay 10 binakabuo. Is that clear? Is that clear? No? Am I making sense, ladies and gentlemen? Am I making sense?
Yes, sir.
So what's the end product of your glycolysis? We're able to produce your pyruvate. Okay? We're able to produce your pyruvate. Okay? So pyruvate, siya, no? For every glucose that you produce, kanina focus lang mo aning reaction. So for every glucose that you use, you're able to produce 2 pyruvate, 2 NADH. By the way, this NADH is present in cytosol, ha? Okay, glycolysis is occurs in cytosol. And then you're also able to produce 2 ATP. Okay? So pyruvate is a versatile metabolite. So if the body is aerobic, it will be converted to acetyl-CoA. However, if the body is anaerobic, instead, it will be converted to lactic acid or lactate. So when lactic acid accumulates, it will cause sprain, no? Or muscle ache. No? When lactate accumulates in the muscle, it will cause muscle ache. So maubito na class, when you do exercise, of course, your muscles will become anaerobic. So as muscle cells produces pyruvate, instead of entering the Krebs cycle, it will actually be converted to lactic acid or lactate. For that reason, when lactate already accumulates in the muscle, it will cause muscle ache. However, let's say, for example, you don't do a lot of exercise. So therefore, your muscles will actually be aerobic. So your aerobic muscles, so it will convert pyruvate to acetyl-CoA. So in acetyl-CoA, no? So once the acetyl-CoA is already produced, it will then enter the Krebs cycle. So in other words, pyruvate is the key metabolite that lets the, pyruvate is the key metabolite that is the one that can enter the Krebs cycle. Is that clear? Is that clear? So this is how pyruvate enters the Krebs cycle, class. So it undergoes oxidative decarboxylation. So pyruvate undergoes oxidative decarboxylation. So this occurs, by the way, already in the mitochondrion. The activation of pyruvate to acetyl-CoA occurs in the mitochondrion. Therefore, what happens here is pyruvate is removed with carboxyl group, no? And then the acetyl that is left will attach to the coenzyme A, forming now your acetyl-CoA. And because this is oxidative, no? Because this is oxidative, it would need the conversion of NAD to NADH. Okay. So how many pyruvate did we produce from glycolysis? How many pyruvate? We were able to produce 2. So therefore, we were able to produce 2 acetyl-CoA, 2 NADH, and 2 carbon dioxide. Do you follow? Do you follow? Class, am I making sense here? Am I making sense? Hello, are you still here? So afterwards, so once the acetyl-CoA is produced, it will now be able to enter the common metabolic pathway. Okay? Sige. Questions? Questions? Pag nag-loading, nagin mo class. Pag did I get healthy if I continue pa? I cannot see any response from you anymore. So, ani na lang. Okay? So, I will stop. No, it's useless for me to continue anymore. Let's just going to summarize glycolysis. So, again, glycolysis is 10-step. Okay? What step requires ATP? Okay? What steps requires ATP? You have steps 1 and step 3. Do you follow? What commits glucose to glycolysis?
Step 3.
Step 3. What produces NADH? I think I created a note, a table for this in your notes. What produces NADH? Step 6. What produces ATP? What produces ATP? Step 7 and
Step 10.
Step 10. Okay. So for every glucose that enters the cell, what's our byproduct in glycolysis? You're able to produce 2 pyruvate, 2 ATP, and 2 NADH. Do you follow? And then what is the significance of pyruvate, class? It is the one that is converted to acetyl-CoA. Do you follow? It is the one that is converted to acetyl-CoA, and acetyl-CoA undergoes Krebs and etc. Do you follow? Do you follow? So I hope that you somehow get that connection. Okay, like what I've mentioned, I don't require you to memorize the steps of glycolysis, only the Krebs cycle. As long as you're able to see the connection between glycolysis and the common metabolic pathway. Okay? Sige. Questions so far? So with regards, class na wala pa nag-calculate, no? We weren't able to calculate how many ATP did we produce yet, no? We haven't calculated how much ATP did we produce. So I will just give it to you as a recorded lecture. Okay, I don't think you would be able to understand further if I will continue discussion. So with regards to how I was able to get 30 and 32 ATP, I will give it to you as a pre-recorded lecture. There's a question here. This was start to glycolysis and ends with ATC. Yeah, that's correct. So glucose catabolism starts with glycolysis and ends with ATC. You mean kaning 30? Kaning 30 or 32, Miss Balanay? No? You mean this? No, the amount of ATP that is produced? Yeah, that is starting from glycolysis down to ATC. So this is the amount of ATP that is produced per glucose molecule. So I will explain that in a pre-recorded lecture. Because if I continue now, it's mura gyapon ko pre-recorded, no? Kung mag-continue kog discuss ani, mura gyapon ko nag-record. Kaya ending, dili na magyapon mo makasabot. Diba? So yeah, it involves Krebs cycle, Jade, no? It still involves Krebs cycle. Because like what I've mentioned, yeah, it still involves Krebs cycle. Because like what I mentioned, your pyruvate is the connection between carbohydrate and the Krebs cycle. So it is still related to Krebs cycle. Do you follow? Do you follow? Okay? So related na siya. So iyan mo ng connection. All right? So any other questions? Any other questions?